Welcome back, friends. Suppose we have isolated a DNA and we want to check it whether we have successfully isolated it or not. Or suppose we have performed a PCR experiment and want to analyze that PCR product. So, to visualize the DNA, we need to do this agarose gel electrophoresis. Well, as the name suggests, it uses electric current and gel for separation of molecules. वो कहते हैं ना नाम में क्या है ना नाम में ही सब कुछ है. As the name suggests, agarose gel electrophoresis, we are using electric current and gel for the separation of molecules. Whenever we apply electric current to gel, molecules on the basis of their size gets separated. Well, not only agarose gel, but we can also use polyacrylamide gel even for DNA also. Yes, you have heard right. We can also use polyacrylamide gel to visualize DNA also. We'll discuss about it later. As of now, let's focus on agarose gel electrophoresis. So first what we do is that we weigh agarose and then dissolve it in either TAE buffer or TBE buffer. And then we need to heat it. Since when we need to till when we need to heat it? Until the solution gets transparent. And after that we need to check whether the temperature has lowered or not. We can do it by touching a hand on the flask, and if it's tolerable, say it has reached 40-50 degrees Celsius, then we can cast it to the casting tray of electrophoresis unit. But here there is an interesting tale. Actually, ETBR is that key due to which we can visualize DNA, and this ETBR can be added in several steps. First, when the agarose gel solution has reached 40 to 50 degrees Celsius, then we can add ethidium bromide. And so, after the gel has been cooled and ETBR is added, we pour it into the casting tray. But before that, depending upon the number of samples, we use that type of comb. Suppose we have 6 samples, so we can use a comb which will make 8 rectangular holes or slots. If our sample number is more, then we need to use another kind of comb. So we put the comb and then we pour our agarose gel. And then we leave the solution for 15 to 20 minutes at room temperature. Then we simply put the casting tray in electrophoresis unit and then add TAE or TBE buffer depending upon our requirement and availability. That gel should be completely wet with that buffer. The gel should not be dry. The buffer should rinse the gel actually. As DNA is negatively charged, so it is kept at the negative side only so that it can migrate towards the positive side. So, on the rectangular holes or slots, one puts its DNA, the sample mixed with dye, and then we know that DNA is negatively charged due to presence of phosphate groups in it. So, it migrates towards the positive pole. Now, the rate of migration of a molecule depends on the concept of friction, which we have studied in our middle school. Basically, small DNA molecules will experience little frictional drag from solvent and gel molecules, so they can migrate easily, they will migrate rapidly. However, large DNA molecules will obviously encounter more friction, so it will migrate slower, and ultimately the result will be that the electric current will distribute the DNA fragments according to their sizes, and the largest one will be near the top and the smallest near the bottom. This is how we can visualize DNA. By the way, as I have just said now that we can add staining dye ETBR in agarose gel before casting, but we can also avoid adding ETBR that step at that step. Instead, After adding DNA in those slots and running it completely, at last we can also add that ETBR, for which we need to soak that it in ETBR for 30 to 40 minutes. So this is how Agarose electrophoresis looks like. But there are some important key points in this technique that when we prepare agarose gel, we need to keep in mind that what percentage of agarose we are measuring. For example, if we prepare 0.5% agarose, that is say zero point 0.5 gram in 100 ml of TAE, it will have large pores and we can use it for molecules in the size ranging from 1 to 30 kb. Generally, during practicals, we use 1% agarose gel. For further smaller size, we use more percentage of agarose gel. Also, in the starting, I had mentioned that we can use polyacrylamide gel here also. Yes, we can use that if we want to separate much smaller DNA molecules, say in the range of 1 to 300 base pairs only. Then we can use 40% polyacrylamide gel, and due to it, it will have extremely small pores, which can be used to separate much smaller DNA molecules too. Well, we generally add ETBR, but we all know that it's carcinogenic. It can intercalate with the bases and can cause cancer. It is a mutagen, okay. But it has also limited sensitivity. Suppose one say that we will be careful and we can use ETBR even though it's mutagenic, but a bigger problem is that it also has limited sensitivity. If our DNA has less than if there is less than 10 nanogram of DNA, then we cannot visualize our DNA. In that case, we need to use different fluorescent dyes that stain DNA red, blue, or say green. These sensitive dyes are able to detect bands that contain even less than 1 nanogram of DNA. Last but not the least, we can estimate the size of DNA molecules via various mathematical relationships. So this was all about agarose gel electrophoresis. If you have any query, you can ask me in the comment box.